We tend to think of animal behavior, especially the mating stuff, as just raw instinct, pure drive.
Right. It seems pretty straightforward on the surface.
But when you actually dive into what's really fueling those urges,
Mhm.
you find this incredibly complex, surprisingly adaptable system.
Absolutely.
Yeah.
Yeah, what looks like a simple biological imperative is actually this fascinating dance between, well, the animal's internal hormones and everything happening around it, the environment.
And that's exactly what we're plunging into today. We've got a stack of, well, really compelling scientific reports here
Uh-huh.
that really dig into how these two things, hormones and the environment, work together, how they shape sex drive, sexual behavior,
Right.
across a huge range of species, too.
We're talking the nitty-gritty, right? Neuroendocrine factors,
Yeah.
environmental influences, the whole picture.
Exactly, the whole complex picture. So our mission today for everyone listening is to sort of pull out the most important bits of knowledge from these sources.
Yeah, get past the jargon. We
Right.
want to reveal some of the honestly unexpected ways animal sexual behavior is driven and just how flexible, how malleable it can be.
Okay, let's get into it. Let's unpack this hormonal orchestra, as the sources sort of frame it. Testosterone. That's probably the first one that pops into everyone's head, right? Especially for males.
Definitely. It's the classic association.
But it's not as simple as just like having high levels floating around.
No, not at all. It seems there's more of a threshold effect,
The threshold, meaning,
meaning an animal needs a certain baseline level. Enough testosterone to kickstart things and keep sexual activity going.
okay,
But once you cross that threshold, simply having more testosterone doesn't automatically mean more sexual behavior. It's not linear like that.
that's really interesting. So it's not just more equals more. And the sources even mentioned these kind of cycles, rhythmic ups and downs in sexual behavior in male rats,
Yeah, that's fascinating.
even after their initial encounters with females, when their testosterone levels have sort of settled down.
Exactly. It's like there's an internal rhythm, maybe, that isn't just about the immediate amount of testosterone swimming around.
So what does that tell us about the mechanisms underneath?
Well, it really highlights this difference between what they call organizational versus activational effects of testosterone.
Organizational,
Right.
activational. Okay, break that down.
Okay, so organizational is like the early exposure during development. It sets up the brain circuits for male sexual behavior. It builds the hardware, if you will.
Got it. Lays the groundwork.
Precisely. Then activational effects happen in adulthood. That's when testosterone and its metabolites actually act like the key. They unlock those pre-wired circuits that were built earlier.
Ah, okay. So the groundwork is laid early, and then current hormone levels just flip the switch on the established system.
Pretty much, yeah.
But it's not just the levels in the bloodstream triggering it. The sources talk about local hormone processing in the brain being vital, converting testosterone right where it's needed.
Yes, and this is where it gets really cool, I think. It's about specific enzymes in specific brain regions.
Like aromatization.
Yes, aromatization, that's converting testosterone into estradiol. Yes, an estrogen by the enzyme aromatase.
Wait, estradiol, isn't that usually considered a female hormone?
It is, but that's the twist. In brain areas like the medial preoptic area, the MPOA, this local conversion to estradiol is absolutely critical for activating copulation in many male mammals and birds.
Wow. So male animals need a bit of, well, female hormone action in their brains to actually initiate mating.
In a manner of speaking, yes. Locally produced estradiol in that specific brain area drives male behavior. It really blurs those simple binary lines we sometimes draw.
That's wild. Okay, and then there's the other pathway, 5 reduction, converting testosterone to DHT.
Right. Dihydrotestosterone, or DHT. That seems more involved in the physical mechanics, the peripheral stuff. Genital reflexes needed for the act itself.
So 2 different pathways from the same starting hormone doing different jobs. Brain motivation versus physical function.
That's a good way to put it. Aromatization in the brain for the drive and the act, and 5-RE reduction more peripherally for the, well, plumbing.
And these hormones, T and its metabolites, they're actively changing things at the cellular level, aren't they?
Oh, absolutely. They bind to specific receptors, androgen receptors, estrogen receptors inside the neurons.
Oh.
In those key brain regions we mentioned, like the MPOA, the medial amygdala, the BNST.
And that binding does what?
That hormone receptor complex then goes into the cell's nucleus and actually changes gene expression.
It tells the cell to make different stuff?
Essentially, yes, different proteins,
Right.
which ultimately changes how that neuron functions, how it talks to other neurons. It physically alters the circuitry to facilitate sexual behavior. It's a very direct influence.
Mind-boggling. Tiny molecules rewriting cellular instructions to drive behavior. And the sources touched on motivation, too. Testosterone linking up with dopamine pathways. The reward system.
Yes, that's a crucial piece. It suggests sexual behavior isn't just a reflex. There's a strong motivational component, linked to pleasure and reward. And testosterone likely helps fuel that drive through its interactions with dopamine. It reinforces the behavior, makes it feel good, makes the animal want to do it again.
Adds that layer of wanting it. Makes sense. Okay, let's switch gears. What about the female side of things? Estrogens and progesterone are the big players there, right?
Absolutely. For many species, certainly rodents, they're key. They regulate receptivity, that's the willingness to mate, often shown by postures like lordosis.
That distinctive arched back posture in rats, yeah.
Exactly. And also proceptivity.
Perceptivity, that's more like the solicitation, the initiating behavior.
Precisely. Like flirting or signaling readiness. Receptivity is the yes. Proceptivity is the maybe or the invitation leading up to it. And these are very, very tightly linked to the estrous cycle, time for maximum fertility.
A very clear hormonal cycle driving behavior. But the sources pointed out this isn't quite the same in primates, including us.
That's a really important distinction, yeah. Hormones definitely still play a role in primate female sexual behavior, but the link isn't nearly as rigid. It's not solely dependent on the cycle.
What else comes into play then?
Social factors, cognitive factors.
Yeah.
They have a much bigger influence. The whole picture is just, more complex, less directly tied to immediate hormone levels.
But even in those species where it is more hormonally driven, there are specific brain areas controlling it. The VMH kept coming up, ventromedial hypothalamus.
Yes, the VMH is critical, particularly a specific part, the ventrolateral bit, the VMH-VL. That's key for lordosis.
And specific cells within that.
They've even identified specific neurons called VMH-VL PR positive neurons, basically. Neurons in that area that have progesterone receptors, and they are absolutely essential for female receptivity.
Wow. So specific. And MPOA, which we talked about for males, that's involved in females too.
It is. Which is interesting, right? It seems to be involved in female sexual motivation and even mate preference. So there's definitely some overlap in the brain regions used by both sexes for sexual behavior.
Shared neural real estate, just activated differently depending on the hormonal context.
You got it. Different hormonal cocktails acting on similar underlying structures.
And estrogens. They do more than just sexual behavior in females, don't they?
That's true. The sources link estrogens to maternal motivation, too, influencing the drive to care for offspring.
So it highlights how these different behaviors, sex, and parenting might be interconnected hormonally.
Yeah, suggesting maybe a shared evolutionary path or just efficient use of the same signaling systems for related reproductive functions.
Now, this was a bit surprising to me, the mention of androgens, typically male hormones playing a role in female sexual behavior. How does that work?
It seems to be a nuanced role, not the main driver, perhaps. Studies using specific drugs, SARMs, that activate androgen receptors in female rats. They found it could boost preference for males, increase perceptive behaviors.
So it did have an effect.
It did, but here's the catch. It was mainly seen in females who were already sexually experienced and had less than optimal levels of their main hormones, estrogen and progesterone.
So it's not driving the bus, but maybe fine-tuning things.
Exactly. That's a great analogy. It suggests androgens might sort of tweak the motivation or the expression, especially when the main hormonal signals aren't quite optimal, like adjusting the volume knob, not turning the radio on.
Okay, that makes sense. Estrogens and progesterone are the main conductors, but androgens can pop in for a little supporting solo to adjust the sound.
Something like that, yeah.
All right, let's move on to prolactin. Now, most people, myself included, probably think milk production, lactation.
That's the primary association, for sure.
But it seems to have a much more complicated relationship with libido. What's the surprising angle here?
Yeah, it's definitely not just about lactation. It seems to have these dose-dependent effects on male sexual behavior, sometimes even opposing effects, based on studies in rats.
Dose-dependent, so different amounts do different things.
Exactly. At normal physiological levels, it seems involved in, normal reproductive function. There's even a temporary spike after ejaculation in males.
Oh, what might that do?
It's possibly linked to that feeling of sexual satiety, or maybe even plays a role in a short-term facilitatory effect, though that's still being explored. Point is, it's not simply an inhibitor at normal levels.
Okay, But when levels go wrong, when they get too high, hyperprolactinemia, or HP, the sources called it.
Right. That's where it gets complicated. Moderate HP, so moderately high levels in rats. It was actually linked to increased mounting frequency, more attempts.
More attempts, so higher motivation.
Maybe. But it also led to difficulties with intromission and ejaculation, so the drive might be there, but the execution is impaired somehow.
Like wanting to drive but having engine trouble.
Kind of, yeah. But then, if you get chronic and severe HP, really high levels for a long time,
Yeah.
that has a clearly inhibitory effect.
How so?
Low testosterone, erectile dysfunction, reduced libido, the works. It really shuts things down reproductively.
Yeah.
So there seems to be this optimal window for prolactin.
A delicate balance, then. Too little might be bad, but too much definitely throws the system off. And the mechanism involves dopamine, right? How do they interact?
That's the current thinking, yes. It's a
Yeah.
bit of a feedback loop. Dopamine normally acts to inhibit prolactin secretion from the pituitary gland.
Okay.
But then prolactin itself can influence dopamine activity back in the brain in areas like the nucleus accumbens and the MPOA, those crucial zones for sexual behavior.
So they influence each other.
Constantly. And the idea is that high sustained levels of prolactin might actually downregulate dopamine activity in those reward and motivation circuits, which could contribute to the sexual inhibition you see in severe HP.
Wow. It's this intricate dance between hormones and neurotransmitters. And prolactin's role isn't just about sex itself, it's also linked to social bonding, like oxytocin.
Indeed. That study on cotton-top tamarins was fascinating.
They're monogamous primates, right? Cooperative parents.
That's them. And they found a positive correlation between prolactin levels and things like huddling together, grooming, contact affiliation, and sexual behavior within pairs.
So higher prolactin went along with more closeness and more sex.
Yes, and interestingly, prolactin levels even tended to be similar between partners in a pair bond.
That sounds a lot like what we hear about oxytocin.
It does. It mirrors oxytocin's role in social bonding quite closely. It suggests prolactin might be another piece of that puzzle, helping maintain pair bonds and social connections, maybe acting through similar pathways or influencing the same brain areas.
It's incredible how these hormones wear multiple hats, depending on the context, the species. Now let's talk stress. We all know stress messes with us, and it seems it has a pretty big impact on animal libido too, via glucocorticoids, cortisol and the like.
Oh, absolutely. That link is very well established. The HPA axis, hypothalamic pituitary adrenal axis, or HPI, hypothalamic pituitary interrenal, in fish and amphibians. That's the body's main stress response system.
The stress alarm.
Exactly. And it triggers the release of glucocorticoids, cortisol in primates and fish, corticosterone in rodents, birds, reptiles.
And how do these stress hormones affect sex drive?
Well, a short-term stress response can be adaptive, fight or flight. But chronic stress, or really severe stress, leading to elevated glucocorticoids, that's generally bad news for sexual function.
Why is that?
It's essentially the body saying, look, times are tough, survival is paramount, now is not the time to be investing energy in making babies. Reproduction gets put on the back burner.
The sources had clear examples, like prenatal stress affecting offspring development.
Massing effects, yeah. Programming the stress axis differently, influencing sexual differentiation.
And chronic stress in female rats reducing their receptivity, making them less willing to mate.
Right. And that finding in ewes was interesting too, where cortisol seemed to inhibit receptivity, the willingness to mate, but not their attractiveness to males or their perceptive soliciting behaviors.
What does that selective effect suggest?
It suggests a potentially fine-tuned mechanism. Maybe even under stress, the female is still checking out potential mates, assessing options.
Okay.
But the actual act of mating, which is risky and energetically costly, is suppressed until conditions improve. It's a clever way to prioritize resources and safety.
Prioritizing survival over reproduction when things get tough makes biological sense. And the ways stress inhibits reproduction, it's not just one thing, is it? It hits the system from multiple angles.
Definitely multi-pronged. Glucocorticoids can suppress the whole HPG axis right from the top, reducing GnRH release, which then reduces gonadotropins, which reduces sex hormone production.
So it shuts down the command chain.
It can, yeah. They can also act directly on the gonads, the testes, and ovaries to inhibit steroid production right there.
What?
And they can even interfere with how the sex hormones actually work at their target tissues in the brain and elsewhere. Plus, stress impacts peripheral functions, like reducing nitric oxide synthase in the vagina, affecting physiological readiness for sex.
So it's attacking the system at multiple levels simultaneously: central command, production lines, and even the final execution.
Pretty much, yeah. It's a comprehensive shutdown strategy when chronic stress takes hold.
And the social context matters here too, right? Like social status influencing stress levels.
Absolutely. Social status is a big one. Subordinate animals often experience higher chronic stress, higher baseline glucocorticoids, which feeds back into suppressing their reproduction.
And sex differences too.
Yes, there can be sex differences in how the HPA axis responds to chronic stress, potentially influenced by the sex hormones themselves. The whole social hierarchy adds another layer of complexity onto how stress impacts reproduction.
Okay, so the picture emerging is it's incredibly interconnected. Hormones are central, but they're constantly being tweaked, modulated by everything happening around the animal, especially the social world.
Exactly.
And speaking of other players, our sources mentioned a few more hormones and neuropeptides that help fine-tune this whole system.
Yes, beyond the headliners like testosterone and estrogen, there's a whole supporting cast. GnRH, for example, gonadotropin-releasing hormone.
That's the master switch at the top of the HPG axis, right?
What is it? Kicks everything off. But it also seems to have direct effects in the brain, acting as a neuromodulator itself, influencing behaviors like lordosis directly.
Interesting. And its counterpart, GNIH.
Gonadotropin inhibitory hormone. As the name suggests, it acts as a brake. It inhibits gonadotropin release, countering GnRH, and it might also directly inhibit sociosexual behaviors. In quail, for instance, it seems to inhibit aggression and sex, possibly by increasing neuroestrogens.
So GnRH is the gas pedal, GNIH is the brake for the reproductive
So
system,
got it.
and kisspeptin, that sounded crucial too, especially for puberty.
Kisspeptin is hugely important. It's a key upstream regulator of GnRH, absolutely essential for puberty onset. Without it, puberty doesn't happen correctly.
But it does more than just start puberty.
Yes, it seems to have ongoing roles, direct roles in things like mate preference and lordosis, in female mice, for example. And its activity is itself regulated by ovarian hormones, showing it remains integrated into the adult system.
Okay. And we can't forget oxytocin, the so-called love hormone. Its roles go beyond just birth and lactation, right?
Oh, far beyond. We already touched on its role in social bonding, like in those tamarins.
Right.
And that study in rams was interesting, showing oxytocin administration could actually improve sexual performance and reduce stress responses during mating tests.
So it might facilitate positive social and sexual interactions, maybe by reducing anxiety?
That's a possibility, yeah. It's involved in a whole suite of pro-social behaviors.
And its cousin, vasopressin, or vasotocin in non-mammals, that sounded more involved in things like aggression, social recognition, but
Right.
also pair bonds.
Exactly. Vasopressin vasotocin is another key modulator of social behaviors across vertebrates. Aggression, territoriality, recognizing familiar individuals, and yes, forming and maintaining pair bonds in some species. It often works in concert with, or sometimes in opposition to, oxytocin, depending on the context and species.
So many different chemical messengers involved.
It really drives home the point. Libido isn't just one thing. It's not a single switch. It's an emergent property of this incredibly complex neuroendocrine network.
Right.
A network that's constantly integrating all these internal signals, hormones, metabolic state, with external signals from the environment. And that Table 1 in our source materials does a nice job summarizing these key players and their diverse roles.
It really is like a biological symphony, isn't it? All these hormones and neuropeptides playing their parts, conducted and constantly adjusted by the environment.
That's a perfect analogy.
So let's really focus on that conductor, now the environment itself. How does it directly shape this hormonal orchestra and ultimately libido? The social context seems huge.
It really is.
Yeah.
Social interactions are a primary source of information for animals, information they use to adjust their hormonal state and behavior. Think about dominance hierarchies.
Okay. The pecking order.
Right. Dominant individuals, the top dogs, often have higher levels of androgens testosterone, or 11-ketotestosterone in fish. And sometimes, though not always, lower baseline stress hormones, lower glucocorticoids.
And this relationship goes both ways.
It seems to. Winning fights, achieving dominance, that can actually boost testosterone. It's like a biological reward for social success. This fits into the biosocial model or the challenge hypothesis.
Success breeds success, hormonally speaking. What about the losers, the subordinates?
Well, being subordinate often comes with the opposite hormonal profile. Lower androgens, higher chronic stress hormones. And that combination typically suppresses libido.
Which might actually be adaptive for them.
It could well be. Why waste energy and risk injury trying to reproduce if your chances are low due to dominant rivals? Suppressing libido might be a way to conserve resources and avoid costly conflicts until perhaps their social situation changes.
Makes sense from a survival and energy perspective. Don't pick fights you can't win, especially over reproduction.
Mhm.
And it's not just about fixed status, right? The immediate presence of others matters too. Partners, rivals.
Oh, absolutely. For males, just being exposed to receptive females can trigger a rapid surge in testosterone, primes them for courtship, for mating.
Gets them ready.
Exactly. And conversely, the presence of male rivals, that often ramps up testosterone and aggression. That's the challenge hypothesis in action, then the body preparing for competition.
And I was really struck by that example you mentioned, the prison effect in male turtles. Limited access to females leading to weird behaviors.
Yeah, that study was quite something. It really highlights how crucial appropriate social cues are. When those normal stimuli are absent, or the social environment is highly artificial, sexual behavior patterns can become disrupted, even anomalous like same-sex behaviors in those turtles. It underscores the need for a species-typical social context.
And we already touched on pair bonds and affiliation prolactin, oxytocin helping maintain those connections and correlating with sex in tamarins.
Right, and even things like social touch, grooming, or just the experience of being reared in a group versus isolation, all these social factors can have profound and lasting impacts on the development and adult function of these neuroendocrine systems controlling behavior.
So the social world is this constant feedback loop, shaping hormones, which shape behavior, which then influences the social world again.
Precisely, it's dynamic.
Okay, let's shift from the social to the physical environment. Things like light cycles, temperature, how do they cue the reproductive system?
The physical environment provides really fundamental cues and constraints. Photoperiod day length is probably the most studied environmental cue for reproduction.
Because it reliably predicts seasons.
Exactly. For many animals, especially outside the tropics, day length is the best indicator of upcoming seasons. So they use it to time their breeding cycles, synchronizing reproduction with the best time of year for food availability, favorable temperatures for raising young, etc.
And how does the body read the day length?
It involves the pineal gland, which produces melatonin, mainly at night. The duration of melatonin secretion tells the brain how long the night is, and thus how long the day is.
Okay.
This melatonin signal then influences the HPG axis, often via another pathway involving TSH, thyroid-stimulating hormone, and thyroid hormones acting within the hypothalamus, ultimately controlling GnRH release. It's like the environment setting the master biological clock for reproduction.
So the seasons set the stage via light, and the hormones follow suit. And it's not just reproduction affected by light, right?
No, photoperiod influences a lot more. Metabolism, immune function, even mood in some species. It's a really powerful fundamental environmental signal, even for animals that aren't strictly seasonal breeders.
Fascinating. Okay, what about temperature? That seems critical, especially for some animals.
Absolutely critical, particularly for ectotherms, the cold-blooded animals whose body temperature depends on the environment.
How does it affect them?
Well, their entire metabolic rate is tied to temperature, growth, activity levels. The temperature size rule describes how many ectotherms develop faster but end up smaller at higher temperatures. And temperature directly influences their activity periods, including when they can be active for mating.
And for endotherms, like mammals and birds, we regulate our own temperature, but does external temperature still matter?
It does. We have thermoneutral zones where we function optimally. Extreme heat or cold can be stressful, diverting energy away from things like reproduction. High temperatures, for example, can negatively impact sperm production and viability, reducing fertility. And libido itself can certainly take a hit during extreme weather.
And temperature can interact with other environmental factors too, like pollutants.
Yes, that's a growing concern. Temperature can influence the effects of endocrine-disrupting chemicals, potentially affecting sex determination in some reptiles and amphibians where temperature plays a role, or exacerbating the impact of pollutants on reproductive physiology.
It's also interconnected. Physical factors, chemical factors. What about the habitat itself? The quality of the place an animal lives? Degradation, fragmentation. How does that play in?
Habitat quality is hugely important. Degradation often means less food or lower quality food, impacting nutritional status, which is critical for reproduction.
We'll get to nutrition in a sec, yeah.
And then there are chemical pollutants, pesticides, heavy metals, endocrine disruptors, which can directly mess with hormone systems, affecting development, fertility, and behavior.
And fragmentation, breaking up habitats.
Fragmentation causes a host of problems. It restricts movement, making it harder to find mates or access resources. It reduces gene flow between populations. It increases edge effects, exposing animals to different conditions or predators. all of which can increase stress levels and negatively impact reproductive success, including the drive, the libido needed to initiate it. It's often a cascade of negative effects.
It really paints a picture of animals constantly navigating their surroundings with their hormones and libido acting as this sensitive barometer responding to all these pressures.
That's a great way to think about it.
Okay, let's talk about food. Nutrition. That seems absolutely fundamental. How does the nutritional landscape shape libido?
It's foundational. Reproduction is incredibly energetically expensive. Think pregnancy, lactation, raising young.
Huge energy demands.
Exactly. So an animal's nutritional state acts as a key signal to its neuroendocrine system. Do I have the resources to pull this off?
And if the answer is no,
If the animal is in negative energy balance, burning more calories than it's taking in reproduction generally gets put on hold. This can mean delayed puberty in young animals, longer periods of infertility after giving birth, postpartum estrus, disrupted ovarian cycles, lower conception rates. The body prioritizes basic survival and maintenance when energy is scarce.
so if you're struggling just to stay alive, making babies is off the table. Makes sense. And it's not just about total calories, is it? The type of nutrients matters too.
Oh, absolutely. Quality matters as much as quantity. Adequate protein is essential for growth, for making hormones themselves, for fetal development. Too much protein can sometimes cause issues too, like urea toxicity affecting fertility in ruminants.
Okay.
Dietary fats are crucial for energy storage and, importantly, as precursors for making all those steroid hormones, testosterone, estrogen, progesterone.
The building blocks.
Exactly. And then micronutrients, minerals like zinc, manganese, selenium, copper, vitamins. They're essential cofactors for enzymes involved in hormone synthesis, tissue integrity, immune function, everything really. Deficiencies can definitely impair reproduction.
So you need not just enough fuel, but the right mix of fuel and building materials. A balanced diet is key for libido too.
Pretty much. It sounds like a Goldilocks situation, not too little energy, not too much, and the right balance of nutrients. And body condition score, or BCS, is often used as a practical indicator of this nutritional status and energy reserves. Both very low and excessively high BCS can negatively impact reproduction.
And nutrition interacts with everything else, doesn't it? Like lactation being even more demanding or poor quality forage in a degraded habitat, making it harder to get those nutrients.
Precisely. It's all interconnected. The endocrine system essentially acts as this gatekeeper, monitoring metabolic signals like leptin, insulin, glucose levels, and only giving the green light for optimal reproduction when resources seem sufficient and the animal's in good enough shape. It's a biological risk assessment based on energy stores.
We've touched on stressors throughout, but let's focus specifically on how stress itself acts as a direct modulator. We know it releases glucocorticoids. What are the key ways these stress hormones then suppress libido?
Right. Stressors, whether they're environmental like extreme weather or pollution, or social like conflicts or isolation, they activate that HPA or HPI axis.
Uh-huh.
And the resulting glucocorticoids, as we discussed, inhibit reproduction at multiple points in the HPG axis. They can suppress GnRH, reduce gonadotropin sensitivity, directly inhibit gonadal steroid production, and interfere with sex hormone action in the brain.
A multi-level inhibition.
Exactly. And prenatal stress is particularly potent, capable of permanently altering the development and programming of both the stress response system and the neural circuits underlying sexual behavior in the offspring.
Those effects can last a lifetime.
They can, yes, and it's important to distinguish acute versus chronic stress.
Right. A quick scare versus long-term pressure.
A short-term acute stress response is generally adaptive. It helps the animal survive an immediate threat. But chronic stress, where the HPA axis is activated repeatedly or for prolonged periods, that's usually maladaptive. The sustained high levels of glucocorticoids can cause widespread problems, including reproductive suppression.
And animals don't always react the same way to repeated stress, do they? Habituation, sensitization.
Exactly. Sometimes animals habituate, meaning the stress response lessens with repeated exposure to the same stressor. Other times, they become sensitized, showing a stronger response. And in extreme chronic cases, the HPA axis can even become downregulated or exhausted, leading to different physiological problems.
But the general outcome of chronic stress on libido is usually negative.
Generally, yes. Sustained glucocorticoid elevation tends to suppress the HPG axis, leading to lower sex hormone production and reduced libido. Stress can also have indirect effects by causing weight loss or poor body condition, or even affecting secondary sexual characteristics that influence attractiveness. The duration and intensity of the stress are really key determinants of how badly reproduction is affected.
This has been incredibly insightful, looking at the hormones and the environment, but The way we study these things matters too, doesn't it?
Hmm.
Lab versus wild, captivity versus nature, that context must influence what we find.
Oh, hugely. That's a critical point. We absolutely have to consider the research context.
How do things differ, say, comparing hormone profiles between wild and captive animals?
While often, though not universally, captive wild animals tend to have higher baseline levels of glucocorticoid stress hormones compared to their truly wild counterparts, though capture itself is stressful, so getting a true wild baseline is tricky.
Right.
Domesticated animals, interestingly, often have lower baseline glucocorticoids than wild ones, possibly reflecting adaptation to human environments and reduced predation pressure. Behaviorally, captive animals often show different time budgets, sometimes with more abnormal stereotypic behaviors and fewer complex natural behaviors compared to wild individuals.
So the environment of captivity itself imposes stress and changes behavior.
It certainly can. The stressors inherent in captivity confinement, artificial social groups, lack of natural foraging opportunities, human presence can lead to potentially lasting changes in endocrine function, immune responses. We often see reproductive suppression in newly captured wild animals as they struggle to adapt.
Does the type of captivity matter, like a zoo versus a more natural enclosure?
Yes, species respond differently, but generally the quality of the captive environment makes a big difference. Enclosures that are larger, more complex, and mimic the natural habitat more closely tend to result in lower stress levels and more natural behavior patterns compared to restrictive, barren conditions.
And this carries over to how we interpret research findings from different settings, lab versus field.
Absolutely. Each setting has strengths and weaknesses. Lab or captivity studies offer high experimental control. You can manipulate specific variables precisely.
The strength is control.
Right. But the weakness is the artificiality. The environment might induce stress, the behaviors might not be natural, the animals might not be representative of wild populations, and the findings might lack ecological relevance.
Okay. And field studies.
Field studies offer high ecological validity. You're seeing the animal in its natural context, but the weakness is lack of control. It's hard to isolate variables. There are logistical and ethical challenges in sampling, especially invasive sampling like blood collection.
And the act of touching them can skew hormone results.
Exactly. Capture stress is a major confounding factor for hormone measurements in field studies. That's why non-invasive methods collecting feces, urine, saliva, hair, feathers are increasingly valuable, though they need careful validation, and you have to consider time lags between hormone production and excretion.
So the bottom line is we can't just take a lab finding and assume it applies directly in the wild, or vice versa.
Precisely. Captivity is not a neutral background. We need to be really cautious about extrapolating findings. The ideal approach is to integrate data from both contexts, always acknowledging the limitations of each method. It's about building the most complete picture possible from different angles.
Okay, so pulling all these threads together now, the hormones, the environment, the research context, what are the big patterns we see across the vertebrate family tree? Are there common themes, surprising divergences?
Well, there's a fascinating blend of conservation and divergence. The basic hormonal machinery, the HPG axis, steroid hormones like T and E2, stress hormones is ancient and conserved across vertebrates. But how it's used, how sensitive it is to different cues, and how rigidly behavior is tied to hormones, that varies enormously.
So give us a quick tour. Mammals.
We've covered mammals quite a bit, the key roles of T, E2, progesterone, prolactin, glucocorticoids, strong modulation by social factors, and, in many, photoperiod. Birds also rely heavily on testosterone, especially its aromatization to estrogen in the brain, for male song, aggression, and sexual behavior. The challenge hypothesis is well supported in many bird species. Photoperiod is often the dominant environmental cue for timing reproduction. Interestingly, high levels of neuroestrogen can sometimes be inhibitory to male sexual behavior in birds.
Reptiles.
Testosterone and progesterone are important for male behavior. Temperature is often critical, not just for activity, but sometimes even for sex determination. The social environment can be surprisingly potent, like inducing those anomalous same-sex behaviors in turtles we mentioned. Captivity effects can vary, perhaps less severe in large semi-natural enclosures.
Amphibians.
Neuropeptides, like vasotocin, the amphibian version of vasopressin, are key modulators of social and sexual behavior. They're highly vulnerable to habitat fragmentation affecting movement and breeding, and extremely sensitive to endocrine-disrupting chemicals in their aquatic environments, which can wreak havoc on development and sex differentiation. Temperature is also a major influence.
And fish. They seem quite flexible.
Fish show remarkable social plasticity. In many species, social status can change rapidly, accompanied by swift changes in the HPG axis GnRH, gonadotropins, and the fish-specific androgen 11 ketotestosterone. Neuroestrogen roles are complex, potentially inhibitory sometimes. GnRH, kisspeptin, GnIH are all crucial players. And being aquatic, they're highly susceptible to endocrine disruptors in the water. Plus, unexpected roles for hormones like oxytocin are emerging, like its involvement in heart repair in zebrafish.
Wow. So the same basic toolkit, but adapted and implemented very differently depending on the animal's lifestyle and environment?
Exactly. It reflects their diverse evolutionary paths and the specific ecological challenges they face. And that table 2 in the source materials gives a good comparative overview of how different environmental factors impact these groups.
It really highlights how libido isn't predetermined, but emerges from this constant integration of internal state and external world.
Absolutely. Environmental factors, social, physical, nutritional, stress are continuously tweaking the neuroendocrine system, modulating hormone levels, changing receptor sensitivity, altering neural circuits. It's a constant adjustment.
And these factors interact, right? Like poor nutrition making an animal more vulnerable to stress.
Definitely. The effects are often synergistic or additive. Photoperiod might set the broad seasonal window for reproduction, but within that window, factors like food availability, social cues, and stress levels fine-tune whether and when reproduction actually occurs.
So the neuroendocrine system is the central integrator, the conductor constantly adjusting the hormonal orchestra based on all incoming information, trying to make the best reproductive decision in the current circumstances.
That's it precisely. Libido isn't a fixed quantity. It's a flexible, plastic, context-dependent expression of an individual's reproductive strategy at that particular moment.
Okay, this has been a truly deep dive. As we wrap up, what are the main sort of take-home messages you want listeners to remember?
I think a few key things stand out. First, the multi-hormonal control, it's rarely just one hormone. Second, the dissociation sometimes seen between hormone levels and behavior, brain sensitivity, and local metabolism are critical. Third, the profound and dynamic role of the environment in modulating everything. Fourth, the importance of research context lab versus field matters. And finally, this fascinating pattern of conserved systems but divergent implementation across the animal kingdom.
And these insights have real-world implications, don't they?
They absolutely do. For animal welfare, understanding stress effects can help us improve conditions in zoos, labs, and farms. For conservation, recognizing how pollution, habitat loss, and climate change impact reproduction via these neuroendocrine pathways is vital for developing effective strategies.
And maybe even some cautious insights for ourselves.
Well, we always need to be careful extrapolating directly, but understanding these fundamental biological mechanisms of how hormones, brain, and environment interact to shape behavior in other animals certainly offers valuable perspectives on the deep biological roots of behavior in general, including our own.
It definitely broadens the perspective. Are there still big unanswered questions in this field?
Oh, tons. We still need to nail down the precise molecular mechanisms for things like prolactin's dual effects, or exactly how the brain integrates signals from multiple simultaneous stressors. The long-term, potentially epigenetic consequences of early life environmental exposures on adult libido and reproductive function is a huge area. And we always need better, more refined, non-invasive techniques for studying hormones and behavior in wild animals without disturbing them.
So more integration is needed.
Yes, integrating across levels, from genes and hormones to behavior and ecology, using combined approaches, will be key to fully unraveling the beautiful complexity of what drives animal libido.
Well, it's certainly clear that the world of animal libido is far more intricate, far more sensitive to the world around it, and frankly, far more surprising than most of us probably imagine.
It really is. It's this constant dynamic interplay between internal drives and external pressures, shaping one of the most fundamental aspects of life.
A truly intricate dance, and it definitely leaves you wondering, how might these deep biological principles, maybe in ways we don't even recognize, be subtly influencing behavior across the entire animal kingdom? Food for thought.
Definitely food for thought. There's always more to discover about the hidden drivers shaping life around us and within us.